Hello, everyone, and welcome to today's broadcast, Validating and Publishing with Antibodies, How to Avoid Reviewers' Challenges, presented by Dr. Chandra Mohan, a Senior Manager in Scientific Content Training at MilliporeSigma. I'm Dr. Susie Valdez of Labroots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Labroots and sponsored by MilliporeSigma. For more information about our sponsor, visit them at www.emdmillipore.com. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box located on the far left of your screen and type the questions into the drop-down box that appear on the screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing the presentation, just click on that Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Just click on the Continuing Education Credits tab located on the top right corner of the presentation window and follow the process for obtaining those credits. Please join me now in welcoming our speaker, Dr. Chandra Mohan. I will now turn the presentation over to him. Welcome, Dr. Mohan.
Thank you, Susie. Welcome to MilliporeSigma's virtual seminar. Today's topic is validating and publishing with antibodies and how to avoid reviewers' challenge. You might be working for 6 months to a year to get publishable data. You prepare the manuscript with great diligence, submit, and wait for a letter of acceptance. And sometimes it comes back with minor revisions, and at other times it's rejected, which is very disappointing. So today we'll talk about a few simple steps that you can take to improve your chances of acceptance of your manuscript. So before I commence with the main topic, I would like to bring your attention to the fact that Millipore and Sigma are now one company, and Sigma was acquired by Merck KGaA from Germany in 2016. That's our parent company, and as a combined organization, we carry over 300,000 products, including about 90,000 antibodies. We are in 77 different countries, and we have 72 different manufacturing sites throughout the world. Validation of antibodies is a big question. And also a big question is, why do we need antibodies in biological research? Over the past 2 decades, you have noticed that researchers, for safety and regulatory reasons, have shifted from radioactive techniques to more immunotechniques. And as immunotechniques are improved and modified, more and more people are adopting these techniques. Why do we use antibodies? Because we want to identify a target molecule, which is important for us. We want to locate the target molecule within the cell, whether it's mitochondria, whether it's nucleus or endoplasmic reticulum. We want to see where it is located. And sometimes we can use these antigen molecules as cell or tissue type markers. Some of them may be specific only for one particular tissue. Some of them will tell us the physiological stage or the disease state of a tissue. Those are some of the important things. The target molecule can also be used for purification, concentration, or further analysis by help of antibodies. So when you're doing this kind of work and you're publishing, a reviewer can always ask you a question. The marker you have chosen is not exclusively found in that cell or that cell type. And that will be a question for you to answer to the reviewers. So our response will be, don't be too target protein focused. Take, for example, GABA versus GABA transporters. Are they present in the same cell? Many different markers may exist for a particular type of tissue. Now, you can use at least 2 different markers for the same tissue to support your hypothesis. Sometimes the stains you use for the histochemical work may be cell-specific. Nowadays, more and more people are also using mRNAs, messenger RNAs, or microRNAs, which can support your publication to show to the reviewers that you have already checked these things and your choice of antibody is good. Now, first of all, we have to see why antibodies. Antibodies have high binding specificity. And this specificity can be used to identify, isolate, immunoprecipitate, even enrich samples. So that's why we use antibodies in multiple applications. Now, it's not as easy as said that antibody specificity is the one which you should use and all your experiments will be good. It can vary. Antibody specificity does not mean it is very reliable unless you take precautions. Antibody specificity can be affected by multiple factors, such as the concentration of the antibody you use, what's the buffer system you use, is the pH altered, is there presence of any detergents which can affect the performance, how was the animal immunized, when did we collect the blood. If we collect the blood at early phase, the titer will be very low. There may not be enough antibodies. Then if you collect in the middle phase, maybe after 2 months after injection, the antibody titer will be very high. And towards the tail end, maybe 6 or 7 months later, if you go and collect your sample, again the antibody concentration could be low. So those are the factors which can affect your choice of antibody as well as the performance of your experiments. So a reviewer can always ask a question, can make a comment. The buffer chosen to dilute your antibody has detergents, which is known to affect the HRP, that is horseradish peroxidase, and DAB, which is diaminobenzidine, in reaction. How do you respond to something like that? Now, this is a common question they can ask, but you have to know and justify the steps in your protocol. It's not always that one protocol which you obtain either from a textbook or from a publication or from a vendor is the one you have to use. It's not one size fits all protocol. You must adapt the protocol to your experiments, adapt the protocol to what kind of detergents, what kind of reaction buffers you have. So every antibody is different, so you have to optimize each antibody, and sometimes with each different lot you have to do that because titer may be different with different lots. Now, antibody choice can also depend on your application. What type of tissue sample you're using, what type of cells, or the cell lysate or tissue homogenate you're using. So always have the target into your mind. Target consideration is important. Where is your target located in the cell? Is it in the nucleus? Is it in the cell membrane? Then the processing of the tissue will be different. What type of sample is to be used? Also, sample preparation. How do you prepare the sample to keep the antigen intact? If you use very harsh treatment, antigen may be destroyed. If you use homogenization techniques or cell lysis technique, some of the non-specific proteases may come into play and they may destroy your small amount of antigen. So what precautions you have to take? Those are some of the things which are important in antibody choice. Antibody design consideration is important too. What's the purpose here? We want to see whether a monoclonal antibody is better suited for your experiment or is a polyclonal antibody that is better suited for your experiment. What's the epitope sequence? Is the epitope sequence totally buried inside in a tertiary structure, or the conformational change has to be brought about? Those are some of the things in the antibody design you have to take into account. Now, again, the target antigen consideration affects the assay design. Take, for example, with our antibodies, which are used in small amounts, you have to design your experiment well. You have to plan ahead. Ask questions from suppliers and the vendors who are providing you antibodies, colleagues who have used these antibodies in their work. So some of those things are important. It's important to know where the target is. Is it internal target? Is it external target? Is it transmembrane? That means you have to use certain detergents to bring that antigen out of the membrane. How do you do that? Do we have to use protease inhibitors? Do you have to use protein phosphatase or a protein kinase inhibitor in your experiment to maintain the phosphorylation state of your antigen? These are some of the important considerations which you have to take into account when you are designing your experiments. Sometimes the structure of the molecule, structure of your antigen can be important on how do you bring it out so the epitope is exposed where the antibody is going to react with that. So these considerations are built into development of quality and should be evaluated prior to antibody use. So even before you buy the antibody, you should take these things into consideration. On the right side, you see a small book here, which I'm going to put a little bigger image in the next slide. It's called An Introduction to Antibodies and Their Applications. It's about a 100-page book, and we have published this a couple of years back. In 2015, we wrote this book. It's about a 100-page book that provides detailed knowledge about antibody use, theory of antibody, etc. What are the precautions, technical tips? Much information is there. This is an excellent reference source for any lab. At the end of the presentation, I'm going to provide you my email address, and you can request a copy. We provide this free of charge to any researchers. Just ask for it, and this will be highly useful in your lab. Now, this book has a lot of information. It has theory of antigen-antibody reaction. A lot of protocols are provided, troubleshooting guides, technical tips. A lot of watch-outs are there that what not to do, how to avoid certain pitfalls in the experiment. These are the kind of information which is included in this book, which you'll find it very useful. In addition, we also have sample protocols for ELISA, Western blotting, immunohistochemistry, immunofluorescence, etc. And we have many tips and tricks. And we also have a glossary of commonly used terms, and that will be really useful for newcomers in the field of immunobiology. Now, it's important to know what sample you are going to be testing. So even before you purchase an antibody, this is an important consideration. Some proteins are expressed only in certain selected cells or tissues. And so if you are using an antibody, let's say you use a protein kinase antibody, will it react with particular tissue? Take, for example, protein kinase C gamma isoform is present only in brain and nowhere else. So you have to be careful on what tissue you use. If you use PKC gamma on some other tissue, it's not going to give you a reactivity. So nature of the target protein and location of the target protein is important. And the publisher usually doesn't ask because they know whether you use the right material or not. But you should provide sufficient data, either your own data or published data, to show that the target protein is being expressed in your tissue or cells. So this is important. Now also it's important that expression levels can change. Expression levels change usually depending upon the physiological state of the tissues or depending upon the disease state of the tissue. The levels of the antigen can change. So that you have to keep in mind. If there's a very low expression, how much antibody do you use? If there's a high expression of protein in this tissue or in the cells, then you can even dilute the antibody a little bit so that you get more reliable results. Sometimes what happens is proteins may be short-lived or they may be very labile. And how do you study that? That's the important thing. So take, for example, those who are studying the apoptotic cell death. They know that after induction of apoptosis, whole process is over within a few minutes, 20 to 30 minutes, the whole thing is over. And sometimes you may not be able to detect the endpoint. So it's important to design an experiment in a way where you can detect your results properly. The target is detected at the end in a very nice way. The other important thing is the target consideration, as I mentioned. Sometimes the epitope may be on the pre or pro-protein region. So then if the pro region or the pre-region has been eliminated during the activation of the protein, activation of the enzyme particularly, take for example procatepsin. If the epitope is on the pro region and then you are using an antibody which reacts with that particular epitope, once the enzyme is activated, you may not get a reaction. It will be a negative effect there. So those are important. Sometimes the latent or the activated proteins are detected. Take, for example, some antibodies are specific only to the phosphorylated form or only to the non-phosphorylated form, or other protein translational modification, acetylation. ubiquitination, simulation. Those are the other things you have to take into account when you are designing your experiment. Tertiary structure can obscure target. So when you are denaturing or degrading protocols will be needed in that. So there are multiple things you have to test in there. If you are using live cells, then it's recommended that you use an antibody which is not preserved in azide solution. So those are some of the things important. If it's an intracellular or intramembrane location, how do you bring them out? What kind of detergents you have to use? It has to be very mild detergent which does not destroy your antigen. So those are some of the considerations you have to take into account. Now a reviewer can always ask, can you submit data showing that the antibody you use is very specific? So take for example, here we're using trimethyl lysine 27 site. So if you are using histone antibodies and then you are using one very specific for one particular modification site, here it is trimethylated lysine on site 27. How do you show that to the reviewer that you are right? So state-dependent antibodies must have extensive validation. How do you do that? I'm going to show you some of those. First of all, when you buy that antibody, you go to the vendor website, vendor data sheet. They should provide you the validation data. Protocols and control samples are often available from vendors or from your own lab. And so if this is not available, you should use your own control data. These are important things. Here, take for example, we do one protocol which is called the antibody assurance. Our assurance method for testing shows very high specificity of our histone antibodies. Take for example, here's trimethyl lysine 4 on histone H3. So H3K4 Me3, that will be the trimethylated lysine on histone H3. The lysine is at position 4. How do you show that? So when we do these kind of experiments, when you do the assurance kind of experiments, you'll see that only on the boxes, 2 green boxes there, you will see the reaction. That's 2 boxes are for 10 nanogram and 100 nanogram. And you'll see that all the other boxes are empty. It does not react with H3K4. It does not react with the monomethylated. It does not react with the dimethylated. It reacts only with the trimethylated form. So this is the excellence of validation. So we are not saying that we test all antibodies this way, but some of them where it is very critical, particularly with this modification site, we do test our antibodies this way. It's highly specific binding to the trimethylated lysine on lysine 4 on histone H3. Now sometimes what happens is sample preparation is not adequate. It's usually overlooked process. A lot of people don't pay detailed attention to how you're making your sample, how you are making your lysate, or how you are doing your homogenate without damaging the antigen. These are important things. Sometimes the antigen is present in very small amounts. It's low abundance. How do you do that? In that case, you can always isolate the organelles. Take, for example, your material is present only in the nucleus or only in the mitochondria. Then you do fractionation, and we have various different kits available, which you see in the last column here. So this will cause the enrichment of the organelles, and then you can have a better reaction. You can have an adequate signal generated. Sometimes the target is too small. It's a small peptide of a much larger protein. In that case, you use the size exclusion filtration. You can see on the last column we have provided Emicon ultraside exclusion spin filters. And sometimes it's obscured in the plasma membrane or in another organelle inside the cell. In that case, we have what we call as proteoextract kits, and then a particular type of fraction can be collected and analyzed. And if the signal is very weak because the binding is weak, then you can use some of the other things like subcellular fractionation you can use. You can reach concentration of proteins, and we have the concentration systems available. So these are some of the tricks you can follow or techniques you can apply. Now, before you begin to plan your experiment, check if what kind of antibodies will be best suited for your experiments, whether it's a monoclonal antibodies or whether it's a polyclonal antibody. If it's a large antigen, it will have multiple epitopes. In that case, a polyclonal antibody may be better suited for the reaction. If it's a small antigen and has fewer epitopes, or only one epitope, then chances of better reactivity comes with the monoclonal antibody. So those are some of the things we have to take into consideration when you're doing these experiments. Now, there's a big controversy on our discussion on whether to use monoclonals or polyclonals. The polyclonals have certain advantages and monoclonals have certain advantages. Take, for example, polyclonals, they are easy to make. They give you a better signal. They have multiple epitopes. There are more applications, and they are better suited for denatured proteins. Whereas disadvantage is what happens if the animal die. Then you'd have no source of animal producing antibody. And that if you inject a new animal, it may take a few months to get that. Lot-to-lot variations, because if you bleed the animal at a certain time and then 2 months later, you'll have a variation in the lots. Different bleeds will give different results, and sometimes you can have non-specific bindings. So my recommendation, or our recommendation is you plan your experiment well, assess your antibody needs, order more than one vial at a time so that you can avoid lot-to-lot variation in the antibody. Now, same thing happens with monoclonals. Actually, the reviewer comment on the polyclonal could be, repeat your experiment with a monoclonal to be confident of your polyclonal. So it's not a bad idea, but not necessary. You can always choose a polyclonal made from a shorter peptide instead of the whole molecule. Take a short peptide which is specific only for that antigen. And then you can also do multi-applications. So you can do Western blot and immunohistochemistry, or Western blot and flow cytometry in the same experiment if the antibody works in those applications. So at least do 2 applications. So that gives confidence in your own data, and the reviewers like that. Now, the monoclonal antibodies have certain advantages. They have high specificity. They have less batch-to-batch variation. They bind only to one antigen. And the hybridomas can be maintained, and you have the infinite source of antibody. But the disadvantage could be they are labor intensive, they are more expensive, they are limited applications, and they may not work in all species. And sometimes the epitope could be affected by chemical treatments. So those are some of the disadvantages. So you have to make that decision before you start your experiments. So sometimes the reviewer can ask, the error bars or the standard deviations are disturbingly large on your ELISA data. How do you answer that question? So one thing you have to do is you have to lock down your protocol, and you can order all your antibodies from the same vendors from the same lot. And you can confirm with confirm your experiment using a monoclonal antibody by increasing replicates. So instead of doing 1 experiment, 2 experiments, just do maybe 4 or 5 repeats of that. So it may give you statistically better results. Now, antibody validation is necessary, but not very well defined. So why worry about validation? Now, a couple of years back, 2014 to be precise, Amgen scientists started working on some 54 different targets based on the publications. And they wanted to repeat these experiments to take it further so that they can do some drug development studies. And what they found was out of 53, they could repeat only 6. That's like 11% success. So there was a big issue raised that why they could not repeat this even in the industrial settings. So multiple publications from academia and industry have raised this issue of reproducibility, that's the specificity of antibodies. And so based on that, many different publications came out, and some of them were from industry and some of them were from academic labs questioning the specificity and reproducibility of antibodies. Even our own company, we have published 2 different publications here, what you see on the top, light blue, evolving strategies and applications and further consideration of antibody validation, where we bring out some of these issues with antibodies. Now, based on that, there are a lot of recommendations have been made, It's like multiple approaches to improve reproducibility are being explored. They are even talking about establishment of a database housing unique antibody IDs and their development information. Something like a central library of clones, something like you have for the cells with the ATCC. The funding agencies and publishers should ask for more details on the development and validation of antibodies. We should be able to provide more trainings for the users of antibodies and the interpretation of those data. And that's why we wrote that book on the antibodies, Introduction to Antibodies and its biological applications. And so the improvements in validation has to be done by the developers of antibody, whether it's you're getting that antibody from a vendor, commercial vendor, or a friend or a different lab. You should be able to validate that antibody before putting those into your experiment. Now, I'm not going to go into this detailed slide. What it shows is whose job it is to validate antibodies. Ultimately, of course, you are the one who is publishing it, so your job is also to make sure that the antibody used is proper. And as supplier of antibodies, the commercial sources are supposed to be responsible to make sure that they provide you with a good product which works in all your experiments, or at least in the tissue samples or into the applications which they are designed for. So this is an important thing. Now, a couple of years back, the Science magazine did a webinar series, and what they did was they talked about the precision and accuracy. So how do you demonstrate precision and accuracy? It's like a target. If it is a sensitive issue, it's a sensitivity issue, that's a precision. Reproducibility is also precision. Specificity means it reacts only with one particular epitope or one particular antigen, that's accuracy. You should be able to repeat that. It should be application specific. So if we say it works in immunohistochemistry, it should work in that. That's also accuracy. So it should be sensitive, it should be reproducible, it should exhibit specificity for the antigen, and it should give you specificity in that particular application. Those are the things discussed in that. So a reviewer can ask you, I'm not convinced of your antibody specificity. They're not convinced of the specificity of the antibody. So how do you convince them? Use two or more different techniques to verify specificity. Maybe it's a Western blot and immunohistochemistry, or immunocytochemistry and ELISA. Use two or more antibodies to different immunogens or regions for the same antigen. Test against state-dependent controls, including knockouts, which we are going to talk about a little later. So use of knockout models are becoming more and more popular now. So we do at least two different types of testing. At MilliporeSigma, we do at least two types of testing to validate our antibodies. Sometimes even more than two testings are done on the antibodies. So a reviewer's comment could be, antibody failed in Western blotting but seems to work in immunohistochemistry. That means the antibody appears to be of low quality. How do we overcome that? It is not uncommon to have antibodies which work in one application but not in others. It's common, especially in monoclonals. Check with the vendor. Even before you buy the antibody, check with the vendor if there is any particular protocol you have to follow. The epitope may be blocked in that application. Confirm using different antibodies against the same target or to different epitopes on the same target. Those will be some of the consideration. And once you include that data, it's not that you have to provide all data in the same publication. Sometimes you can send supplementary data to the editors, which they will be passing on to the reviewers. So those are some of the things you need to take into consideration. Now, antibody and assay validation is part of a good experimental design. So there are multiple things you can do depending upon the target and depending upon the antibody. It's like a peptide inhibition assays are done, microarrays are done, multiple application validations are done, and take for example here Western blotting and flow cytometry. At least two different kind of testing is done on validation. You always use controls. So those are some of the good experimental habits which you can use. At MilliporeSigma, what we do is for all our new antibodies, which for last several years now, we have been doing testing in at least 2 most relevant applications to the target. It could be Western blotting, immunoprecipitation, immunohistochemistry, flow cytometry, sometimes chromatin immunoprecipitation, as the case may be. We also purify our antibodies in a very robust way. We want to ensure the specificity and lack of any cross-reactivity of antibodies. So those are some of the things which we do at MilliporeSigma. Take, for example, some of our validation include enhanced validation. We call it EVs or enhanced validation. Now here in this particular case, take for example, we have treated samples with kinase-specific activators or inhibitors to verify specificity. We do phosphatase treatment to remove that phosphate group so the reaction does not come in. Sometimes the blocking experiments are done with antigen peptide to eliminate all signals. Sometimes dot blots are done to confirm specificity. And we also check for the correct subcellular localization, or by following a certain treatment, if the molecule moves from one location to the other within the cell, those kind of things can be verified. In addition to that, what we have been doing is, for some of our antibodies, we do the genetic validation with RNAi or siRNA knockdown. The CRISPR-Cas9 techniques are highly used for gene editing and creating knockouts. We can compare data from wild type and gene knockout cells. Sometimes gene function can be suppressed that way. So here, take for example, we are getting a reaction for using the anti-ATRX here, Western blot. It gives you one band, single band, whereas at 2 different concentrations, when you're using the siRNA, we don't get a reaction. That means they blocked. So now this shows the specificity of the antibody. In addition to that, sometimes what we do is we do what is called as orthogonal validation. Here we compare the immunological techniques with the level of mRNA using RNA sequence protocols. Now, expression of mRNA correlates very well with the protein expression levels. Now, here on the left, we have the Western blot for the antibody to vimentin, anti-Vim. And on the right side, we have the mRNA expression. So here is the PLA2R1 expression. Wherever we see the expression, it correlates well with the mRNA level also. Additionally, we also use multiple antibodies against the same target to confirm protein presence in that particular sample type. So these are some of the important considerations for you to know when we are picking antibodies, when you are buying antibody from a particular vendor. You should have a proper control data. You can have a positive result, but where is the negative result? Some tissues may not express, or sometimes you have a negative staining. How do you do those? You check for the vendor's data. If we are not provided, you have to do it your own. What type of controls we do? Lot-to-lot variation, how do we avoid that? It is important to note that any validation cell have good controls. For example, if you are doing immunohistochemistry, we should show both positive and negative staining. When you check vendor data sheet, always check for the controls. How robust is the data? These are some of the important things you have to keep in mind. The type of controls could be positive control, negative control, sometimes the isotype controls, state-specific control, whether it's a phosphorylation, acetylation, and application-specific reagent controls, whether the signal is being produced or not produced because of certain things present in this experiment. Either then we do test with no primary antibodies, no secondary antibody. Different applications we tested. So those are some of the important things. And also one important thing is when you compare antibodies, check vendor data. Price is the last thing you want to check. You want to get quality material. And most of the antibodies vendors are pretty competitive in pricing. But quality is something which you have to always check. Check to see what type of sample was tested. Was it tested on cell lysate or tissue homogenate or particular type of cell line? That's a good data. Sometimes the data by when generated only by using a purified protein or recombinant protein. And this is supposed to give you a result. But is that something which you can use, or you trust that in real live conditions where you're using cell lysates or tissue homogenates? Other thing you want to check is, see if a verification of the presence of antigen is provided by ELISA, or whether there is more in-depth data like immunocytochemistry, flow cytometry, immunohistochemistry, etc. are done. So those specific application data become very important in your decision-making process. Also, cross-application validation is important because that gives an indication on how robust the antigen-antibody reaction is. If you get very weak reaction in one and very strong reaction in other, okay, you can live with that. But if you get very weak reaction in everything else, then you have to really make a decision whether you can use that antibody or not. Now, vendor data is important. Take, for example, here is a typical example. I won't say that I'm not claiming that all our data sheets look like this, but most of them look like this. A lot of testing goes behind each antibody that we produce. Here we have data on Western blotting, immunohistochemistry, immunocytochemistry, flow cytometry, immunoprecipitation. So some of the antibodies are tested in multiple applications. Not that all antibodies are valid in each application. Some of them will work only in certain applications. But if they work in as many applications as possible, we try to provide that data to our customers. Now, other important thing is what you do when you submit your manuscript. Careful experimental design is important, and that can eliminate a lot of reviewers' concern. Determine the best application for your research need. What type of sample is being tested? What's the immunogen species and design? And at the end, of course, if you want good technical support from a company, don't forget technical service. Sometimes you need help in the middle of your experiments. So a live help in this area will be very valuable. Now, some of the major concerns from the reviewers are incomplete information provided. So if you want reviewers to speed up the process, you should always provide complete name of the antibody, species of the immunogen, whether it's a human, mouse, rat, zebrafish, whichever species the antigen was derived, you should know that. And you know that, but you not put it in the manuscript, so reviewer does not know that. Antigen used to raise antibody. What was the dilution used? Any references you have for the validation? If it's a monoclonal antibody, always include a clone number. If you're providing a catalog number and a lot number for the antibody, that's a plus point for the reviewers. And now you're ready for publication. The publication is a peer-reviewed journal. It involves a complex process. It involves reviews, very critical reviews by people who are very knowledgeable in that area. You can make your reviewers and editors' life easy. Remember, reviewers spend at least 1 to 2 hours reviewing your manuscript, and they don't get paid for it. They are doing just in the name of science. So golden rule will be respond completely, respond politely. Even if the comments are, okay, you have to repeat these experiments, don't think that you are not going to do it, because reviewer is very knowledgeable. They have reviewed a lot of papers. They have reviewed a lot of publications. And this publication is going to be a permanent record of your scientific work. So respond completely, respond politely, and respond with experimental data, and that makes editors, that makes reviewers, and that makes you also happy. And that was my last slide, and thank you very much for listening. And my email address is here, chandra.mohan@emdmillipore.com. If you'd like to receive a free copy of our publication, Introduction to Antibodies and Their Applications, please send me an email, and I'll respond back within the same day and send you a copy. Thank you very much.
Thank you, Dr. Mohan, for that informative presentation. And we'll now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click that Send button. Our speaker will follow up with your questions via email. So let's take a look at some of our viewers' questions. Dr. Mohan, Western blotting is very commonly used procedure for immunodetection of proteins, but it's very time-consuming. Can you give us any tips on how to save time when performing Western blotting?
This is a great question. Unfortunately, there are no shortcuts in the steps involved. However, what we have done is we have designed a particular system, we call it SNAP ID system, to streamline your Western blotting and immunohistochemistry experiments. The concept is very simple here. A vacuum-driven flow of blocking, antibody, and washing solutions reduce slide and membrane handling. We use vacuum to actively drive reagents through the membrane. That means a lot of less shaking, a lot of less dipping, pouring, waiting, etc. are avoided. And instead of hours, you can finish your multiple slides or multiple blots within about 30 to 45 minutes. Also, this technology promotes antigen binding and thorough washing, enabling you to better optimize your Western blotting applications or Western blotting conditions. So what you can do is, if you need more information on SNAP ID, just go to our website, emdmillipore.com, and type in the word SNAP, S-N-A-P, and I.D. and you'll get a lot of information about this particular system.
Thank you for that. And sometimes a reviewer may not understand all of the complications, and if I believe that my data is good, why not just send it to another journal who will publish it? This
can happen when submitting a manuscript. You can always suggest one or two names of scientists who are experts in that field, or you may even indicate not to send to a particular reviewer because you think that your work conflicts with the other person, and then he may be biased in his opinion of your manuscript. So you can always do that. You can always suggest names. Editors usually oblige with that kind of request. It's important to consider that a publication is a permanent record, and others will refer to your publication for years to come. Journals will select only knowledgeable reviewers. For you and others, the quality of publication is also important. Now, sometimes what happens, you think that the reviewer is asking too much work to be done, and at that time you decide that you can send it to another journal. Sometimes the chances are that the same reviewer who is an expert in that particular field get your manuscript again because of his or her expertise, and they will reject it again and even be upset. So best thing is design your experiment well, do the experiment well, and try to answer as many questions as possible. Sometimes you want to put the same data in two different publications. If you're going to do that, tell the reviewers, tell the editors that you have additional data if needed, but you're going to put it in two different publications to make it manageable.
Great suggestions. And Dr. Mohan, a viewer wants to know, are there any drawback in designing knockout experiments when performing antibody validations?
You know, knockout experiments and knockdowns are very powerful negative controls. But even though they are very good controls and very reliable, but it does not provide the confirmation of binding. Also, in some cases, the cell lines against a specific gene may exhibit reduced viability because that gene is important for survival of the cell. So what happens is you knock out that gene, the cell may not survive for too long. Hence, we always emphasize more than one validation technique. You can have a knockdown experiment, but also have at least one more validation done on those samples.
Thank you. And our final question is, how can we get a copy of your publication, Introduction to Antibodies and Their Applications?
Now, as I mentioned a little while ago, this is a very popular book, and we printed a lot of copies, but unfortunately we ran out of all of them, so it's currently out of print. However, if you email me at chandra.mohan@emdmillipore.com, I'll send you a PDF version the same day. So please remember that email address which I had on the slide, chandra.mohan@emdmillipore.com, and send me an email and I'll send you a PDF version same day. Anyway, thank you.
Dr. Mohan, thank you again for your presentation and thank you for your important research. I'd also like to thank Labroots and our sponsor, MilliporeSigma, for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through January 2019. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.